I've got it.
Okay.
So shall I introduce you?
Sure, why not?
Okay, so welcome everyone to the second portion of the structural biology class. So we're moving on from these silly X-ray things into more serious techniques, NMR. For those of us with PhD in NMR, you know, you can never game the other tribe. But anyway, so I am your official instructor of record, but we're lucky enough to have Dr. Hans Vogel, who is a very preeminent NMR structural biologist, who recruited me actually to this program. He is a Royal Academy member, I believe, and one of the very early pioneers in structural NMR and a leader in Canada in this field. So we're really lucky to have Dr. Vogel here. He's going to be leading this section. I'll be on the sidelines. And then at the end, I will pick it up and we'll talk about the applications of NMR at the end. But anyway, so I'll be here and serving as a lead TA to Dr. Vogel for this section.
Thanks very much, Dr. Lewis. And I'll try to share my screen here. Let's see here. You guys can all see that? All right, good. So let's try to get started then. So this is not the very first time that I actually teach this course. I have actually been teaching in Biochemistry 551 probably for the last 20 years or so. And the section of NMR, when I was coordinator of the course, of course, was much bigger than the section on protein crystallography. But unfortunately, a protein crystallographer has snuck over and grabbed control of the course, and now the majority of the course is on protein X-ray crystallography. So we're going to have to make do with a limited number of lectures, and so don't look at old lecture notes from previous courses. Last time I taught this course was in Calgary, was in 2019. I've also taught parts of this course in other parts of the world. And so the material that you have in front of you in part is similar to what I had in previous courses, but of course much reduced. And I've also added a little bit of new material as I started putting these lectures together. So, so there are three protein structures displayed on this first introductory slide. And most of you will recognize that this protein over here has what we call a beta sheet structure. There's multiple strands in here. And if you look at the arrows, they give you the direction of the strand. So this arrow points in a different direction than that one. So this is an anti-parallel strand mostly. And this protein, of course, has no beta sheet structure. This is a completely alpha helical structure. And you see this, for example, is a long uninterrupted alpha helix in this protein. Now, there's very few proteins that are exclusively beta sheet or exclusively alpha helix, and most proteins have mixtures of alpha helix and beta sheet. And you see a slightly bigger protein here, where in red is indicated the alpha helices, and in blue you see there's a beta sheet here. And this is one domain of the protein, and this is the other domain of the protein. And in this domain, you see there's another beta sheet here, and in both cases, that beta sheet is surrounded by alpha helices, and there's also an alpha helical linker that links this domain and this domain together. And this is a pretty rigid and fixed structure where the two domains sort of sit in a fixed orientation with respect to each other. Now, the reason for showing these three structures is that they were all determined by NMR spectroscopy in my laboratory. And so this should just convince you that NMR is very capable of determining structures of proteins, but it does so in solution. And that's already a major, major difference when you think about protein X-ray crystallography, because of course they need protein crystals. And of course, if I want to be critical, I can say that a crystal is, of course, not the natural environment in which a protein does its work. And so this is one of the reasons that a lot of people are still quite enthusiastic about using NMR spectroscopy, because being able to work in aqueous solution really allows you to change conditions to conditions that exist inside the cell, or conditions that exist outside of the cell, or change the temperature, or all these sorts of things. And that also happen in the normal life cycle of a protein. The other reason that I like to show this diagram, you actually see here what we call a ligand usually. So proteins do all kinds of things. And sometimes proteins interact with one another, but many times proteins bind certain things. And when these things are much smaller than the protein and not made up of amino acids, we call those things ligands usually, sometimes cofactors, depending on what they do. And in this case, this is actually a rather peculiar thing that binds here, which we call a siderophore. This is a bacterial iron chelator. And if you look closely in yellow, let me just put the highlighter on here. If you look closely in yellow, you can actually see the iron sitting in that siderophore. And so this iron, this ferric siderophore complex is bound by this protein, and then it's actually taken up into bacteria. So this is one way in which bacteria acquire iron. Now this protein is about 60 amino acids long, so it's a very short protein. It's actually a protein domain, so it's cut out of a larger protein. But many proteins are folded in various individual domains, and you can solve the structures of those individual domains and then later on stitch them together. This is also an individual domain of another protein that is significantly larger. But in that case, this is actually the only folded domain within that protein. The rest of the protein, which is at least as long as this, is actually completely unfolded and does not have a well-defined secondary or tertiary structure. So this protein is about, or this protein domain is about 125 amino acids long. And this one is about 300 amino acids. And this is the largest structure that we have ever solved by NMR spectroscopy in my laboratory. And so I'm saying this already here because there is a limit to how large the protein can be that you can study by NMR. So on the one hand, you have the advantage that you work in solution, which makes it very easy to change solution conditions and mimic what happens in the cell. On the other hand, you have a limit when you want to do complete structures to how far you can go. That's not to say that you cannot study larger proteins by NMR. People like Louis Kay, who is an absolute world leader at the University of Toronto, he studies proteasomes, things that are 600,000 molecular weight by NMR, but then he uses other techniques and he doesn't really solve the structure, but he looks at other properties, mostly dynamic properties of such large proteins. And later on in the course, I'll introduce you to the techniques that we use as NMR spectroscopists to look at very large proteins, trying to find out what they do and how fast they do. All right, so let me try to place NMR a little bit into context, in the context of various techniques that we have, and also in the context of the different applications of NMR. And that's what most of this first lecture of today is actually all about. So NMR is part of this large group of biological spectroscopy. And many of you will have already seen some of this in the biophysics course, where certain techniques such as circular dichroism and fluorescence spectroscopy are already being discussed. And so this is spectroscopy in general is one of the most important ways in which we can actually learn about biomolecules that exist in solution. But then sometimes we can also study them while they're in the solid state. And sometimes that solid state is even organized. And then, of course, it's called a crystalline state. And as I said, for protein crystallography, you need crystals. Without crystals, you can't go anywhere. Now, using the various spectroscopy tools that we have, we can actually learn quite a lot about these biomolecules that we are interested in. First of all, we can look at the structure. So I've just talked about protein structure. So we're talking about the 3D structure, what a protein looks like. Is it alpha helical? Is it beta sheet? How are the domains oriented with respect to each other? That's sort of 3D structure. and 2D structure, but also chemical structure. Chemists use NMR to actually look at the chemical linkages and how a compound is actually built up. Now, another thing you can do with spectroscopy is you can look at dynamics. You can look at how fast are things moving through solution, for example. Or you can look at a protein and you can look at how fast is this protein tumbling in solution. Or you can look at, if you have a protein with two domains, how fast are these two domains rotating with respect to each other? So we can learn about the dynamics of things. And of course, when things happen in proteins, there's also an energetic cost to all of this. And this energetic cost, of course, we can also measure with some of these techniques. And one of the other things that we would like to know in many cases is analytical information. We'd like to know actually how much of it is there. Particularly if we have multiple compounds in solution, we'd like to be able to say, oh, there's that much of this, there's so many millimoles of that, and there's so many millimoles of this third compound. And so all these things can be measured by various biological spectroscopy tools. And so every technique that you may want to use has its unique capabilities, but also its unique limitations. I already told you NMR structures can be determined up to about 50 kilodalton proteins. But if you really want to do a structure from scratch by NMR, please don't try for a protein that's larger than 50 kilodalton because you're spending a lot of time and it's going to be a lot of hard work. But if you only want to know if a protein is alpha helical or whether it's beta sheet mostly, then you can do a simple CD spectroscopy. Now, CD stands for circular dichroism spectroscopy. This is a technique that you've heard about in the biophysics course. And so if that's all you need to know, then just quickly run to the CD spectrometer and run a CD spectrum, and you'll be able to quickly tell whether the protein is mostly alpha helical or beta sheet. Now, if you want to really understand a protein, you usually have to use many different biophysical techniques. And so if you really want to know the structure, you really are limited to NMR, X-ray, cryo-EM, the 3D structure. If you want to know about how strong things are binding to each other, some of these techniques in NMR can give it to you, but there's other techniques, sometimes fluorescence, sometimes other spectroscopic tools, sometimes calorimetric tools that allow you to actually get those data in a much more straightforward way. So you either have to, if you want to really understand the biological protein and all its intricacies, you're going to have to use a combination of different experimental techniques. And at least one of these usually has to be a technique with great resolution, NMR, X-ray, or cryo-EM. So what is then the main use of NMR, X-ray, and cryo-EM in the life sciences? And of course, as a protein person, I'm biased because of all the techniques that we know, we really only have three techniques that allow us to do the three-dimensional structure of proteins. And those are X-ray crystallography, NMR spectroscopy, and more recently, also cryo-electron microscopy. I don't know if you introduced to cryo-electron microscopy yet in any of your courses. I hope some people have talked to you about it because it's really the future of structural biology. It was recently in 2017 awarded the Nobel Prize, and there is some really basic but very good information available on the website of the Royal Swedish Academy of Sciences where you can read up on this. Now, the thing is, X-ray crystallography uses X-rays. And if you go into a hospital and they take an X-ray picture of you, that's okay. But you can't go back 5 minutes later and have another X-ray taken, and tomorrow and have another X-ray taken, because it damages your body. And it's the same with the samples. The X-rays eventually destroy the protein in the crystal. And the same with cryo-electron microscopy, because it bombards the sample with electrons, and the electrons are also high energy, so these samples also degrade. The nice thing about NMR is that it actually uses radio waves. Now, there are radio waves around us all the time. The air is full of radio waves, and we all walk around, and we still walk around tomorrow and the day after tomorrow, and so on and so on. So the good thing about NMR is that you can actually completely recover your sample. You put it in the spectrometer, you measure for a long time, and after that you take it out and your protein is still perfectly intact. And then you put it in the fridge or the freezer. And if you want to do some more measurements, you just take the sample out and stick it back in the machine and you measure some more. And because NMR really is a non-destructive technique, it's actually used in all the hospitals, but it isn't called NMR in the hospitals. They call it MRI, magnetic resonance imaging. And the reason they call it MRI rather than NMRI, which is what they should really call it, because it's essentially exactly the same technique, is that the word nuclear makes all the patients very nervous. They immediately start to think about explosions, such as in Fukushima, and all the dangers that go with that. And so they have, in hospital settings, they have eliminated the word nuclear from the abbreviation. So they call it magnetic resonance imaging. But what they are imaging are nuclei, just like when they do spectroscopy on a person, which is possible. I'll show you some pictures of that later. They don't call it NMR spectroscopy. They call it magnetic resonance spectroscopy because, again, in the hospital settings, they want to avoid that word nuclear. So but we're all scientists, and we understand that nuclear magnetic resonance spectroscopy, the word nuclear refers to the nuclei of the atom. And in essence, all NMR work is done with stable isotopes. There's not a single radioactive isotope. There's no radiation. There's nothing dangerous that's going on here. So, it's a very safe technique. And as you know, when you have your MRI picture taken for certain diseases, as far as I know, all patients come back alive out of the instrument. And I think you can repeatedly go into an MRI for follow-up sequences, and every 2 or 3 days, they're allowed to take an MRI of your body, whereas there's a serious limit as to how many X-ray pictures they may actually take of patients. And particularly, as I've learned over the years in working with people in the Children's Hospital here in Alberta, they really want to avoid putting X-rays out for pediatric patients, for little kids. And so this is another reason that MRI is also quite popular in pediatric settings. All right, now a few words about cryo-electron microscopy. This is from this Nobel Prize from 2017. I like putting this up. This looks like something we see a lot in the news these days. So this is actually a virus with proteins sticking out of the surface. I don't think this is the coronavirus because we didn't really worry about it in 2017, but this is a virus that looks like a coronavirus. And so you can see that with cryo-EM, you can actually do much larger structures than with NMR spectroscopy, right? This is a complete virus. And look at this protein here. This is a protein with 1, 2, 3, 4 different subunits. They seem all very similar in terms of structure. And then these 4 combine into some sort of coordinator structure. So again, it might be useful to look at this text. A technique that fires beams of electrons at proteins. So you can imagine that it eventually damages the samples. For decades, biologists have used X-ray crystallography, blasting X-rays at crystallized proteins. And for many small proteins, so we've already talked about the size of the protein, the structure can be solved by NMR. But labs are now all racing to get into cryo-EM, and we're lucky that as of next January, we'll have a new faculty member in the biochemistry group, Dr. Omid Haji-Ghazami, who actually knows how to do cryo-EM. So this will help us forward, also locally in Calgary, towards this future. Now, there's another recent discovery that people will actually mention to you, and this is very recent because this only really happened this summer. And what happened is there is a company called DeepMind, and they have actually tried to solve protein structures without doing any experiments, by just doing computational work. And these are two of the structures that they display on their website. I'll give you the link on the next slide. And you see in blue is the computational prediction, and in green is the experimental result. Of course, like all good scientists, they picked what they call a typical result. But a typical result obviously means you show your very best result and then try to make the world believe that it's a very typical result. So not all predictions work out as well as this one, but many do. And actually, it's quite impressive what they can do today with AlphaFold. And so in your career, you're going to come across people that are going to tell you don't have to do X-ray or cryo-EM or NMR. You just run it through AlphaFold, and now you know what the protein structure looks like. Well, I tend to disagree because in some details, some of these structures can be quite wrong. But for certain purposes, these sorts of structures, these computer predicted structures, can be very useful, right? To gain some initial insight, to design experiments, and then you can maybe later on prove that the parts of the structure that you're really interested in are really oriented the way they are in the prediction. And so you may still have to do experiments to actually put these in. So now how does AlphaFold work? Well, AlphaFold is actually a prediction program. So it predicts the structure of a protein. And what is interesting is since about 2006, well, actually earlier already, because this is CASP7, so this is the 7th time this conference was held. And we're now in 2020, was the last one, CASP14, so that was the 14th iteration. CASP is what they call, is the abbreviation of Critical Assessment of Protein Structure Prediction. So basically, these are people that try to predict the structure of a protein. And then, and somebody has already determined the structure, and then they get together and then they determine how well their prediction actually worked. And you can see that in 2006, well, small amount of proteins were actually predicted correctly. You see that for a long time, there was really not much progress. In 10 years from 2006, different things. And this is the area of artificial intelligence that started then. And in artificial intelligence, people now have what they call neural networks and all kinds of other computational tools. And what they actually did is that, and they looked at the 170,000 protein structures that were deposited in the PDB, and they used that to train this program to actually learn how to determine protein folds from only the sequence. Now, of course, for the gene jocks, this is lovely, right? You just determine the sequence of your gene, and now you can immediately predict what the protein looks like. Giving the sequence, of course, comes directly from the gene sequence. So it's a complicated process. You can go to this website. It's here, deepmind.com/AlphaFold. And these guys are very nice because in July 2021, they made AlphaFold available for free to the whole world. And so anybody in the world can now put a sequence in there and you can actually start to predict. And we've already used it. We've predicted the structure of some proteins. At 5 domains, 4 domains were predicted correctly. The 5th domain was unfortunately not predicted correctly. And that was the regulatory domain that we were most interested in. So we still have to do all the experiments to really do that. But you can see with AlphaFold 2, they're now over 80% correct in their predictions, if their numbers hold up. Now, I'm looking forward to what happens in 2022. But this is going to be really determinant in the future. And people are going to say, well, let's just look at the structure that we can calculate. And then, you know, for many things that may be good enough without doing all the hard work of experimentally determining it. So they have all kinds of tricks that they use. They look at genetics and searches. They look at homologous sequences. They look at 2 amino acids side by side, and because they have so many protein structures in the protein database these days, that they can actually recognize certain patterns, and then they mix all that together. Anyway, it's artificial intelligence. And all you need to know really is protein sequence here, protein fold over here. Really, that's how it works. For the time being, I would consider this a bit of a black box. But if you really want to use this, you have to look at this a bit more in detail, because there is, of course, a certain confidence score that comes with the prediction. So they actually sometimes tell you that, hey, we calculated the structure, but we're not very confident that it's okay. But for some structures, the confidence is pretty high, and you can get away with that. But please, for the time being, remember that X-ray, cryo-EM, and NMR are used to determine 3D protein structures, and such structures are still deposited in the protein database. The protein database is not filled yet with AlphaFold calculated structures, because then we're basically done. It's still considered a predicted structure. And if you really want to do, for example, drug design and all that sort of stuff, which they do in the pharmaceutical industry, you will still need to do experimental work to confirm this. All right, that's what I'm going to say about these other competing techniques. From here on in, I will really only talk about NMR spectroscopy. But NMR spectroscopy is not only used for proteins, it's also used for various other things, as I've already indicated. So NMR spectroscopy really started during the Second World War, when two fellows, Americans, Felix Bloch and Edward Purcell, actually built the first NMR spectrometer and described the principle and showed that it actually worked. And so they were awarded the Nobel Prize for it in 1952. And so it became quickly a very popular tool for chemists, because they realized it would really help them understand what they had actually synthesized, right? So in your chemistry courses, you've already been exposed to some NMR. But then in 1991, a very bright fellow from Switzerland, Richard Ernst, discovered a different way in which to do the experiment, which is now the way everybody does it, and also started to introduce multi-dimensional NMR. In those days, it was two-dimensional NMR, but these days, everybody does three-dimensional NMR, four-dimensional NMR, and some people go even in the fifth and the sixth dimension, which is a bit weird if you think about it, because the world around me is just 3 dimensions, right? Yeah, but for a computer, it doesn't really matter. You can create a 4-dimensional, 5-dimensional space and work in that. Anyway, somebody, Kurt Wittrich, who also was from Switzerland, he started to apply these techniques that Richard Ernst had developed. And in 2002, he got a Nobel Prize for developing NMR for protein structures. But in parallel, there was also this development of MRI, magnetic resonance imaging. And this, in 2003, the late Paul Lauterbur and Peter Mansfield actually got a Nobel Prize from the medical committee, not from the chemical or the physiology committee, from the for the development of magnetic resonance imaging. And so what is NMR spectroscopy then used for? By scientists, but also by clinicians. First of all, chemists were, of course, first down to shoot. As soon as the technique was developed, they started using it for chemical structure elucidation. Protein structure determination did not really start until the 1990s. The very first structures came out in the mid-1990s. There was a lot of NMR spectroscopy going on in those days. I did my PhD on protein NMR, but I looked at phosphorylated proteins and look what the phosphorus groups in the proteins were doing. And we didn't think at the time that we could use NMR to solve complete three-dimensional structures, but slowly over time, that became possible. And as I've said, it's in solution. This is a big advantage. And then once that was developed, people also realized you could use this to look at DNA and RNA structure. And so many DNA and RNA structures are also determined by NMR spectroscopy because some of them just simply don't crystallize. And then oligosaccharide structure determination. This is also a technique or an area where NMR is being very, very powerful. Oligosaccharides are actually more complicated than proteins and DNA and RNA, because DNA is a linear structure, RNA is a linear structure, a protein is a linear sequence of amino acids. But the problem with carbohydrates is they're branched everywhere. So it's a really complicated structural network. And so NMR can resolve all that, though, and the people have looked at oligosaccharides by NMR quite a bit. Now, there's another field that Dr. Lewis and I are very particular to, which is called metabolomics, where you can use NMR actually to see if you can diagnose certain diseases. And you can do this simply by peeing in an NMR tube. And that half a milliliter of that liquid would be enough to stick it in an NMR spectrometer and to see whether you have a certain disease or not, or to predict whether you're going to die within the next half year or not. Prognosis, it can be very serious. In most cases, of course, prognosis is much more optimistic because it predicts that you're going to have a long and fantastically fine life. Magnetic resonance spectroscopy is actually a form of spectroscopy where you put living organisms within an NMR magnet, and you can actually start looking at how much ATP is in their muscle tissue, how much ATP is in their brain, how much phosphocreatine is in their kidneys, and things like that. So this used to be a very active field of research, but it has never led to any real serious clinical applications. So it's not as popular as it used to be some 40 years ago. But these days, magnetic resonance imaging is everywhere. Every self-respecting hospital does magnetic resonance imaging. And so these are all techniques that are NMR spectroscopy techniques. So here you see an image of somebody's brain. This is a picture that I got from a former PhD graduate student of mine, Dr. Peter Lundberg, who was at the University of Linköping in Sweden. And this is what you can see. And the nice thing about NMR is that it looks at nuclei, and the most abundant compound in our body is actually water. And water is H2O. And hydrogen actually has fantastic NMR properties, as we will discuss. And so these guys are only looking at water. Now, of course, there is not much water in your skull, in your bone. That's pretty hard. So you really don't see much definition there. But look at fantastic definition you can actually get inside the brain, which is a tissue that's very high in water content. And you can see even the spine here. You can clearly see what's going on with it. So MRI has made its place in the world and will remain there for a long, long time. There's a variant of MRI called functional MRI, where they can actually see. This is actually looking at a brain from somebody who's had a stroke. And this person is now being tested every week and has to do gripping a ball and squeezing it. And so they try to measure the gripping strength, and then they see in the brain what areas of the brain are actually being utilized to actually do this reaction. And you see, this is 5% gripping strength, and this person is really struggling and encasing a large part of his brain, because what you measure is oxygen consumption, essentially. Oxygen is paramagnetic, that influences the spectrum. But you see, this person gets better and better as time progressed. And after 6 weeks of practicing, this person now only has a small region in the brain, a little bit here as well, left that's necessary to exercise this. So you can actually see from this functional MRI, because this demonstrates that function improves and gets to a more limited area of the brain, it gets more focused, and you can actually then declare that a patient is doing very well. And here, this is MRS, this is magnetic resonance spectroscopy. This is a mouse that goes right in the NMR spectrometer. Right? It sounds funny, but if a person gets into an MRI and comes back out, and the mouse can go in. But in this case, the objective is not to create these images, but to do spectroscopy on the mouse. And this is a phosphorus NMR spectrum, say of the muscle tissue of the mouse. So there's 3 peaks here that come from the 3-phosphate atoms in ATP, and then there's some phosphorylated sugars and other things in there. And this is really important because all cells are energy driven by ATP. And so you can have a good measure here of the physiology of the cell. And this has been a very, very popular technique for a very long time. Now, this is an NMR experiment, but they call it an MRI experiment, right? This is the magnets. This is what we call the bore of the magnet. So this is the hole in which the sample goes. Now, of course, this is a pretty big sample. Big guy like me has to fit in there, right? And it's a bit claustrophobic if they do a scan of your, this guy's lucky because he's got the detector here around his knee, so he only goes in with his knees. But if they look at your brain, they put the detector around your brain to shove you in the middle there, so you're a little bit claustrophobic. It's not an ideal environment. And it's also a bit nerve-wracking because there's a lot of noise that actually happens during this experiment, which for technical reasons at the moment still cannot be avoided. So you go in there and first of all, you don't see anything. And then you hear this da, 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 dum, dum, and you think you're in a war zone or something like that. So it's, but after a while, after half an hour or so, you come back out and then everything's back to normal. So anyway, this is the, and we call this a horizontal bore NMR. And of course, the magnet, the bore in the NMR has to be horizontal so that you can put the patient easily in and out, right? If you use the vertical bore NMR, you would have to wrap a rope around the patient's neck and dangle him into the, that would not be such a good idea because then there would be trouble. So this is a horizontal bore, whereas all the other NMR experiments that we do, we use a vertical bore NMR machine where the bore is actually down the center in a vertical. And the bore is, of course, much smaller because the samples that we use are much smaller than what is actually being examined here. Now, this is an example of a metabolomics experiment. So in this metabolomics experiment, you see this is a sample of urine. So somebody has produced some pee in an NMR tube, and these are different regions of the proton NMR spectrum. Most of this work relies on proton NMR spectroscopy, just like imaging relies on proton NMR spectroscopy. And these are all different chemical compounds. Not all of them are different. For example, here, this is a peak for compound 21. This is another peak for compound 21. These are two different portions of that molecule that have different chemical environments, but they're both part of the same molecule. And you see the same thing here. These three peaks here, they're from compound number 20. And then this peak here is also from compound number 20. But we know the structures of the chemical structures of all these compounds that are in our blood and in our urine. And so we know what the spectra looks like. So it's pretty easy by NMR to look at a spectrum like this, bring in the spectra of all the known compounds that can be in your urine and match them up and say, oh, this must be this must be this must be that. And then the nice thing is NMR is an extremely good analytical tool because you have to look, you can look basically at the intensities of all the peaks and you know what the concentration of that is relative to all the other compounds. So you can say, well, look, there's a small amount of 23. Look how much 21 there is, right? Without giving it a name right now. So this is the power of NMR as well. It's a very strong analytical tool. And that has made it also useful as a tool in metabolomics. So I'm going to give you one example of metabolomics. This is from a paper that my group produced many years ago. This is a project where we worked together with Martin Storr, who was then in the hospital over in the medical side, and his postdoc, Rudolf Schiko. Unfortunately, the project was not extended because Martin Storr moved back to Germany and Rudolf ended up going back to Austria. So we have not followed up further on this particular study, but they were interested in looking at whether we could find out whether we could tell the difference between patients that suffered from ulcerative colitis or Crohn's disease. So these are to typical inflammatory bowel diseases. And in order to do that, we also studied, of course, a control cohort, so healthy people that did not have this disease. And we were fortunate in this particular case that we had both serum samples, so this is blood serum, blood plasma samples, and we also had urine samples. And so we did NMR metabolomics on this. We measured concentrations of 60 different compounds in these 3 different biofluids. And then you do some statistics and you say, can I tell apart the controls from the disease? And you can see here the blue dots here. This is ulcerative colitis versus control. So the green are the controls. This is ulcerative colitis. And yes, you can see they have separate chemical makeup. So the mixture of metabolites is different. And you can actually say, well, these ones are increased and these ones are decreased. And you can do the same thing in urine. This is a sample of urine. I think this is Crohn's disease versus control. Oh no, these are both serum samples, but you can do the same thing in urine. And now there's a group of other metabolites that are increased and decreased with disease. And so these are potentially very powerful methods to start diagnosing disease, because you only need a blood sample or you need a urine sample to actually analyze who may have this disease. And you don't have to do invasive biopsies or other complicated experiments to actually do this. And so this is an application of NMR, proton NMR in particular, that still continues in many laboratories around the world, including mine still, where people look at various diseases. Now, so NMR is useful. So when we want to start looking at NMR as a tool to look at proteins, which is what the structural biology course is all about, we can use the same NMR spectrometer that we use for metabolomics, right, that I just showed you. So what do those things look like? Well, this is a somewhat older diagram from 2004. And you can see here, this is what the magnet now looks like. So this is the outside of the magnet. And this is now the bore of the magnet. So you see the bore is much, much smaller. And it's vertical, right? It's a vertical bore instrument. And if you look closely, there is something in blue here, which is a layer called liquid nitrogen, right? And if you look closely here, this orange stuff, there's a layer called liquid helium. So basically what this is, it's a thermos. Here's my thermos. And I keep my water cold, right? And I'm thirsty. Still cold because it's isolated between the different layers. So the way they make these magnets is you basically have a thermos within a thermos. So this thermos here is filled with liquid helium, and there's then a coil in there to which they run all kinds of currents, and that generates a really large magnetic field right at the center of where this vertical bore actually runs and where the sample is going to go. Now, liquid helium is quite cold, and if you went directly from liquid helium to room temperature on the outside of the magnet, the liquid helium would evaporate pretty fast. And so what they did is they said, well, you know, we just take this liquid helium and we want to keep it as cold as possible. So they put another thermos around it, which is filled with liquid nitrogen. Liquid nitrogen is a little warmer, boils off a little warmer than liquid helium, but it shields the liquid helium from the outside, right? Liquid helium is expensive. Liquid nitrogen is cheap. And so we fill liquid nitrogen once a week. We fill liquid helium once every 3 months. And so that way we have a pretty stable environment. We have to make sure that these things stay in there because these are superconducting metal wires that are in there. And these things are only superconducting when they're at a cold, cold temperature. So that's the magnet, right? So NMR stands for nuclei, a magnet, and R basically for a radio. Now this is the radio, And these are the nuclei. We're going to talk a little bit more about this, but I already said in the introduction, we're talking about the nucleus in the atoms, right? So we talk about 1H NMR, hydrogen NMR. We usually call proton NMR because it's easier to say. So we're looking at the nucleus, the proton in the hydrogen atom. There's only one proton in there in hydrogen because it's the first element in the periodic table. Most elements have more protons in the nucleus, right? Another popular nucleus is carbon-13. Well, it has a larger number of protons in the nucleus. It also has neutrons in the nucleus. So that's what we're looking at. We're looking at the atomic nucleus. So we're looking at nuclei. We use a big magnet. And here, this is actually a cut through this pore, and you see the sample coming in here. And in next week's lecture, I'll actually show you what these samples really look like. And there's really only 0.5 milliliter of liquid or so in there. So if you want to do metabolomics, this can be 0.5 milliliter of urine. If you want to do protein structure, well, then you better purify your protein and get it at a pretty high concentration in there so that you can look at the protein. But you have to communicate with these samples, right? You have to do something to the nuclei that sit in this sample. So the first thing you do is you stick it in the magnet so it sits at the center of that magnetic field. So the magnet does something to these nuclei. And I'll show you in a minute what that is. But at the same time, you need to then disturb that equilibrium, which is where the radio comes in. The radio bombards these nuclei, brings them out of equilibrium, and then we watch how they go back. That's basically how it goes. You need all three. You need nuclei that have the right property, you need a magnet, and you need a radio. All right, this is what our two spectrometers actually look like. So this is our 700 megahertz. So this is a slightly more powerful one. It has a slightly higher field than our 600 megahertz one. And what it says here is NMR is cool. And the reason that I always put that there is that NMR literally is cool, right? Because it's not, these guys are not cool. They're just nerds like me and Dr. Lewis who like to play with toys like this, right? So we're uncool. But NMR spectrometers are cool because they operate at 4 degrees Kelvin, 4 degrees above absolute zero. That is minus 269 degrees Celsius. That's pretty cool, right? Now, liquid nitrogen, some people like to talk about that, is also cool, but not anywhere near as cool as liquid helium, right? It's only minus 196 degrees Celsius, and it's 70 degrees warmer, right? 70 degrees warmer than this. Yet the sample that sits in the vertical bore sits at room temperature, 25 degrees. And in many cases, we run experiments at 37 degrees because that's the temperature of the human body. And so that's representative for what would happen in our bodies. So this is what NMR spectrometers look like. And if you look closely here, there's a red thing here on top. That's where the sample is going to go in. So you got these really big magnets. Of course, we chose a relatively small guy to stand beside it, so it looked even bigger. But the sample really goes in here. So it's a really big instrument, a really small sample that goes in there. And then the sample sits somewhere here in the middle, and then it's bombarded with radio waves. Here, it's difficult to see because this is our fraction collector that we basically use to put the samples in automatically, and it blocks. But there's another hole there, which looks like that hole where the sample goes in. Now, what can you do? What nuclei can you study? Well, look at the periodic table. Essentially, you can study all these nuclei. And they're all different colors because they have different nuclear properties. But if you look at this, everything in the periodic table has something of color on it, which means that in principle, you can study any nucleus in the periodic table of elements. Now, the important ones to us are going to be the ones that are red in this table, because they're the ones that have a nuclear spin of 1/2 of minus 1/2. All the other ones are 1, 3/2, 5/2, 7/2, 9/2, 5, 8. So there's all kinds of numbers. But the ones that have the most useful NMR properties, those are the ones that have a spin of 1/2. So you can simplify this. You can do this. You can say which are the spin 1/2s and which are the what we call quadrupolar nuclei. So quadrupolar nuclei means everything from 1, 3/2, 5/2, 7/2, 9/2. That's all the quadrupolar nuclei. And those are mostly the blue ones. There's the odd one that is green. That's given here because it's got this number 1. That's for nitrogen 14. So that's given here. But there is, in fact, a spin 1/2 nucleus for nitrogen, which is nitrogen 15. So this is the table that you're going to have to remember, because these are the most important nuclei for NMR spectroscopy. And you can see these are protons, carbon 13, nitrogen 15, fluorine 19, and phosphorus 31, right? And they're all spin 1/2 nuclei. The only one that's not a spin 1/2 nucleus is deuterium. Deuterium is, of course, the same as 1H, except that there's a neutron and a proton, so it has different nuclear properties. So it has a different spin quantum number. Now, if you look at this table, this is very important. Because the first thing you want to know how much of this isotope that I want to study by NMR is actually there. And you can see that here, this 1H, 99.9% of the world of hydrogen is 1H. So you turn your NMR spectrometer on, you select the right frequency to communicate with 1H, and all the hydrogens are already for you in the protein. If you want to do carbon NMR, it gets a little bit more complicated because only 1.1% of the carbons are carbon 13. The other 99% are actually carbon 12. Well, carbon 12 is not on the list. Why not? Because it does not have a nuclear spin. And the rules for nuclear spins are given in a few slides at the end of this lecture. You can take a look at that, and I'll briefly mention them again in the next lecture. But we understand very well that this has 6 neutrons and 6 protons, so they cancel each other out, so to speak, and so they really don't give rise to a nuclear spin. It's only when you have mismatched numbers of protons and neutrons that you actually end up with nuclear spins. Oxygen 16 is another case. There's no oxygen on the list here. We can't look at oxygen by NMR very well. There's no spin 1/2 nucleus for oxygen. And here's N15, nitrogen 15, only 0.365% abundant. Yeah, the rest is N14, 99.7% is N14. Well, that's a quadrupolar nucleus, and quadrupolar nuclei are much more difficult to study. And so we rarely do it. And so you can see that if you want to work with these, it might actually be beneficial if you increase that number. If you did isotopic enrichment, and this is what we do a lot in NMR, we start to try to bring this number to 99%, and we try to bring this number to 99%. And in the next lecture, we're going to talk about E. coli and how we use E. coli to make proteins, because E. coli will build the N15 and the carbon 13 for us into the proteins, depending on what we feed them. The other part of the table that's really important is this one, the sensitivity. There's only one nucleus that has a sensitivity that is very close to hydrogen, and that's fluorine, right? This is 100%, this is 83%. This one is 6.6%, so it's not that bad, but look at all of these. They're actually much more difficult to detect than hydrogen is. And so even though you may bring these up to 100% with isotopic enrichment, you still have lower sensitivity than what you get for protons. Protons in the NMR world are king. They are the best nucleus, they're always there, they're 100% naturally abundant, and they have the highest sensitivity of detection compared to any other nucleus. So 1H NMR officially is, of course, hydrogen NMR, but we always call it proton NMR. All right. And then what you do, you just put them in a magnet, these nuclear spins. So
Dr.
these are the
Vogel,
nuclear spins.
Can I interrupt you for just a second? Is the end time officially 1:55? Is that right for this class? So I guess we're up on time for today.
Yeah, we're done. Sorry?
I
Yeah,
think we're up on time for today.
yeah. Yeah, I just wanted to show this. And then this is last thing I want you to look at, but nuclear spins are like compass needles. And when you place them in a magnetic field, they have an orientation. But if you look closely at it, you see that some point to the north and some point to the south, which is a little different than what you see with the compass needle. All right, so that's all I wanted to really say for today. This is where we're going to start off again on Monday, and then we're going to move